Hello and welcome to Lecture 10 in Bio 110. We're going to be looking at the energy transformation mitochondria and chloroplasts part 1. Basically, we're going to be learning about the electron transport chain. But before we go that, let's have a small review of the material that we have covered so far. So from last week, I want you to review the mitochondrial anatomy, basically looking at the mitochondria as a two-membrane component organelle with an outer membrane and an inner membrane. In between the outer and the inner membrane, you have the inner membrane space, and that intermembrane space, it's a place where a lot of molecules are going to be found, particularly protons. You also have the matrix of the mitochondria, which is you can consider it the cytoplasm of the mitochondria. Now, when we think about the process that we have studied so far, glycolysis is going to occur in the cytoplasm. And I will have a video for you here that I found online that has a review of the reactions involved in glycolysis. You can also remember that once pyruvate is being made from glycolysis, it's going to be transported across the mitochondrial membrane where it's going to be oxidized. And that oxidation is going to result in the generation of an acetyl-CoA molecule as well as the decarboxylation of pyruvate to CO2. You're also going to obtain a molecule of NADH. And following that, the acetyl-CoA molecule will go into a citric acid cycle, and those reactions also are happening in the mitochondrial matrix. And I have a video over here for you from the same authors from the previous one in glycolysis describing the reactions of the citric acid cycle. Now, what we're going to review in general, it's that once the citric acid cycle happens, which as you know, it could be called also the Krebs cycle or the tricarboxylic acid cycle, we are going to have a molecule of acetyl-CoA that is going to enter the citric acid cycle. Now, that molecule can come from multiple different biochemical pathways. It can come from glycolysis, or it can be generated from the beta oxidation of fatty acids or the oxidation of amino acids, and that will create some of the intermediates for the Krebs cycle. Now, as we discussed, it's going to happen in the mitochondria, and per acetyl-CoA molecule, you are going to produce high energy electron carriers, NADH and FADH2. You also are going to generate a molecule of GTP, which later, through substrate level phosphorylation, can be converted into ATP. Now, per acetyl-CoA molecule, you will obtain 3 molecules of NADH, 1 GTP, and 1 FADH2 molecule, as well as 2 carbon dioxide molecules. So before we go further, I would like to touch upon another mechanism that is important and is quite regulated in the cell, and that is the mechanism of glucose synthesis. So what we have seen so far is that from a molecule of glucose, you're going to obtain 2 molecules of pyruvate, 2 ATP net, and 2 NADH molecules net. Now, this happens through 10 enzymatic reactions. Of those 10 enzymatic reactions, enzyme 1, the enzyme in step 3, and the reactions in step 10 are irreversible. Basically, the delta G for these steps, it is highly negative compared to the other reactions, and therefore it are basically driven into only one direction. But in cases of need, the cell is able to make glucose to keep it for later use, and that process is called gluconeogenesis. The process of gluconeogenesis bypasses those irreversible reactions by generating a set of enzymes unique to its process. Now, it's going to bypass step 1 by the generation of enzyme A, it's going to bypass step 3 by enzyme B, and it's going to bypass step 10 in 2 different reactions mediated by enzyme C and enzyme D. Now, the reactions that are happening from the intermediates in the middle of both glycolysis and gluconeogenesis use exactly the same enzymes. So from the step of fructose 1,6-bisphosphate to phosphoenol pyruvate, the enzymes for both systems are identical. Now let's take a look to understand what is the control that is at play in gluconeogenesis and the process of glycolysis. So what we're going to see is that in step 3, you're going to have the substrate fructose 6-phosphate, and fructose 6-phosphate is going to be phosphorylated to fructose 1,6-bisphosphate by the enzyme phosphofructokinase. Now, that enzyme is under tight control by the presence of AMP or ADP as well as citrate. So this enzyme is activated by the byproduct AMP and ADP, whereas it is inhibited by the presence of ATP or citrate. On the other hand, the enzyme that catalyzes the reaction from fructose 1,6-bisphosphate to fructose 6-phosphate, that is enzyme B is called fructose 1,6-bisphosphatase, so it is removing a phosphate from fructose 1,6-bisphosphate. Now, that enzyme, it is also under control, but is controlled differently than phosphofructokinase. The enzyme is activated by the presence of citrate and inhibited by the presence of AMP. So we can look at these reactions, and here I matched the reactions by color. So when we're looking at the reaction that is going to phosphorylate fructose 6-phosphate into fructose 1,6-bisphosphate by the enzyme phosphofructokinase, that enzyme, if the cell is lacking energy and therefore you have an excess of AMP and ADP, the enzyme phosphofructokinase will be activated to drive the production of more energy for the cell. But in the case that the cell has a lot of energy, therefore ATP is present and citrate are also present from the citric acid cycle, as we're going to see, the enzyme is going to be inhibited and therefore prevented from moving glycolysis forward. On the other hand, the enzyme that is now going to mediate the dephosphorylation of fructose 1,6-bisphosphate into fructose 6-phosphate, that is fructose 1,6-bisphosphatase, it is having control in a different way. So if the cell is lacking energy and therefore there's a lot of ATP, this enzyme fructose 1,6-bisphosphatase, it is inhibited. Whereas if the cell has a lot of energy and citrate is therefore present, the enzyme is now activated and you allow the gluconeogenesis process to occur. So I am going to leave that step over there, and I'm going to then continue with the topics of oxidative phosphorylation. Here, for your enjoyment, you're going to have the objectives that I would like you to master for this section of chapter 14. And I will leave them here to study on your own. So what I'm showing you over here are two cells, and I want you to take a look at how these cells are organized. What you can see is that the cells have a membrane system that is internal to an outer membrane. So this cell here in the left, you can see the invaginations of the membrane here. And this cell here on the right, it also has a lot of membrane inside the cytoplasmic area. So what we have over here are two prokaryotes, and these are two bacteria that are also respiratory, and they resemble very much the mitochondria. And the fact that they have these extensive membrane systems inside their cytoplasm, it's designed to accommodate the enzymes that are going to fulfill the respiratory reactions that we're going to be talking about today in the electron transport chain. Now, when we look at the entire process of cellular respiration, we're going to be thinking of the cell having the process happening in two different locations with multiple different processes happening together. So to begin with, we have glycolysis, which is going to take a glucose molecule and convert it into two pyruvate molecules. That is going to generate two ATPs by substrate level phosphorylation as well as two NADH molecules. When ATP is transported across the mitochondrial membrane, it's going to be oxidized and decarboxylated. The decarboxylation is going to, per pyruvate molecule, produce one CO2. And also is going to produce one NADH and one acetyl-CoA. Since we have two pyruvate molecules, we have two of each of these molecules. Now, the acetyl-CoA that has been generated from glycolysis can enter the Krebs cycle. And after the Krebs cycle, it's going to generate 2 ATP to substrate level phosphorylation. You're going to have the complete oxidation of acetyl-CoA into carbon dioxide, so 2 carbon dioxides per acetyl-CoA, and you're going to generate also reducing power in the form of NADH and FADH2. Since we have 2 acetyl-CoA molecules going in, we're going to have 6 NADH molecules and 2 FADH molecules generated, as well as 4 carbon dioxides. Now, all the high energy electron carriers are going to donate their electrons in the electron transport chain. And through the process of oxidative phosphorylation, we are going to generate ATP. The ultimate electron acceptor in the electron transport chain is going to be oxygen. So oxygen is going to be reduced into water. Now, the ATP generated through oxidative phosphorylation, it's generated by the enzyme ATP synthase. So what we're going to see is, if everything goes right, a production of about 38 ATP molecules from one glucose molecule. Now, let's take a look at the mitochondrial anatomy to review it one more time. So what we have is the matrix, and in the matrix, which is the cytoplasm of the mitochondria, it's the area where the citric acid cycle and the enzyme ATP synthase is located. So it's located actually at the membrane inside, so the inner membrane area. Now, you have the invaginations of the mitochondria, which are going to generate a large surface area for the respiratory reactions to happen. And you have the outer membrane of the mitochondria as well as the inner membrane of the mitochondria generating the cristae, which are those inner membrane folds. Now, in the inner membrane, you have the localization of the electron transport chain proteins. Now, the inner membrane space, it is the boundary between the the outer membrane. It is the boundary between the cytosol and the intermembrane space, and that is where pyruvate is going to be shuffled to the cell. And at the end, in the intermembrane space, is when you're going to have the high concentration of protons that are going to drive ATP synthesis, and we're going to take a look at that more closely in a moment. Now, mitochondria, since they are the powerhouse of the cell, are going to be located in cells that require a lot of energy. For example, in the left we have cardiac muscle cells, or in the right, surrounding the flagellar core of the sperm, for example, you have all the red mitochondria providing ATP for flagellar movement. And we're going to look at that more closely later. Now, when we think about the reactions that are happening in the mitochondria, especially the reactions that are going to be involved in oxidative phosphorylation, we have to think about it in two terms. Number one, we're going to have the oxidation of NADH and the reduction of oxygen. That is going to have energy in the form of high energy electrons in NADH that are going to be donated to the enzymes in the electron transport chain. Eventually, those electrons will be passed along to reduce oxygen. And at the end, you're going to get NAD+, which is now recycled so it can go back to the citric acid cycle, and water. But at the same time, what we have is the production of ATP. Now, the issue comes is that the oxidation reactions, because you always have an oxidation and a reduction reaction when you pass an electron, the delta G of that reaction is very highly negative, and it is more energy than the positive delta G required for the formation of an ATP molecule from ADP and inorganic phosphate. So what we're going to be seeing is the coupling of the oxidation reactions at the electron transport chain to provide the energy for ATP synthesis through oxidative phosphorylation. So This is a particular case in which both the reactions are coupled, and the end result is the generation of an ATP molecule which has high energy in the form of a phosphate bond in ATP. Now let's take a look to the energetics of oxidative phosphorylation, and I'm going to start by looking at the oxidation and reduction reactions. So the oxidation reaction is going to involve NADH donating its electrons to generate NAD plus and a proton and 2 electrons. And as you can see, that has a delta G naught of minus 158.2 kilojoules. So therefore the reaction is spontaneous. Any oxidation reaction is mirrored by a reduction reaction. So in this case, we're going to have the reduction of oxygen. So half an oxygen molecule plus 2 protons and 2 electrons are going to give you a water molecule. And that has a delta G naught of minus 61.9 kilojoules, which also happens spontaneous. However, when we look now at the oxidative phosphorylation, when we have an ADP molecule plus an organic phosphate and a proton to give us an ATP and water, that reaction has a delta G naught of plus 30.5 kilojoules, which is therefore non-spontaneous. So what the oxidative phosphorylation mechanism has done is to put together these two reactions as shown here in orange. So we're going to have ADP plus phosphate plus NADH plus half an oxygen plus 2 protons, and that is going to give you 1 ATP molecule, NAD plus molecule, and 2 water molecules. And that has a delta G naught of minus 189.6 kilojoules, which makes that reaction spontaneous. Now what we're going to see is that the energy is passed through discrete steps in the electron transport chain. So now, when we look at the processes that are happening in mitochondria, we can divide it according to their stage. So as we discussed earlier, pyruvate generated from glycolysis or fatty acids transported across the mitochondria are eventually going to be converted into acetyl-CoA. Now, acetyl-CoA goes into a citric acid cycle and generates they will be completely oxidized into carbon dioxide, which will then diffuse out of the cell, but you will generate NADH and FADH2. Now, those two molecules are going to be able to donate their electrons to the electron transport chain, shown over here at the top. The molecules that are present in the inner membrane of the mitochondria, of the electron transport chain, will now pass those electrons eventually to oxygen and generating, because of that, an water molecule. So from 2 oxygens, you get 2 water molecules. Now, as those electrons are being passed down the complexes of the electron transport chain, those proteins are also proton pumps. And what you're going to end up having is a high concentration of protons in the intermembrane space, creating a proton gradient shown over here that eventually will be used by the enzyme ATP synthase to make ATP. So ADP that is generated in the cytoplasm will be transported across into the matrix of the mitochondria, and the ATP generated in the mitochondria eventually will be exported out to the cell where it needs it. So again, for you to think about the class and what you have learned, I want you to look at what proteins are missing from the image. So take that, and as a hint, I give you the hint of membrane. Now, let's look at the high energy electron carrier NADH. Because it has a nicotinamide ring, it has been reorganized, it can carry 2 electrons, and those 2 electrons will be eventually donated as well as 2 protons. So those 2 electrons pass to an electron acceptor in the electron transport chain. It's going to be complex 1, and that is going to lead to the rearrangement of the bonds in the nicotinamide ring, and that ends up having now the result of NAD+, which is a charged molecule that has been now completely oxidized. Shown in this image, only looking at the electron transport chain as well as the citric acid cycle, you have the point that the food-derived molecules in the cytosol, glucose, fatty acids, amino acids, etc., eventually are going to bring intermediates into the citric acid cycle. One thing that we'll discuss later is that those intermediates could be in the form of pyruvate, they could be in the form of acetyl-CoA, it could be in the form of alpha-ketoglutarate. Multiple different molecules within the Krebs cycle could then be added, and that is going to then result in the production of reducing power, electrons that are going to be captured in the form of NADH, and not shown here, FADH2. Now, those electron carriers, those high energy electron carriers, are going to be able to donate their electrons to the complexes of the electron transport chain. And as those molecules are passing the electrons in subsequent redox reactions, they're going to eventually be able to transport protons from the matrix of the mitochondria into the intermembrane space, generating a proton gradient. At the end, the proton gradient will be used for the generation of ATP. That is not shown here in this image. However, what is later shown is that those electrons that are being passed are going to eventually be accepted by oxygen to generate water. So at the end of respiration, we get the production of CO2 and water in the reaction. So always, for example, look at the gases that you are exhaling after you breathe. You get vapor of water and you have CO2. So let's take a look at some of the electron carriers that are present in the inner membrane of the mitochondria. We're going to be showing here three major complexes. Though the book doesn't show it, I'm going to show you also another complex that is present. The first complex is going to be the NADH dehydrogenase complex. As the name implies, that is the complex that is going to receive the electrons from NADH. So the complex is going to be reduced, and by a series of redox reactions, it's going to pass its electrons across. And per electron, you're going to have a molecule of a proton being transported across the inner membrane of the mitochondria. Now, you have also present a soluble electron carrier called ubiquinone. It's a highly hydrophobic molecule that is present in the inner lumen of the membrane, so in the hydrophobic region of the membrane, and is able to shuttle electrons from the NADH dehydrogenase complex to the second complex, or actually we're going to call it the third complex, that is going to be the cytochrome C reductase complex. So as the ubiquinone molecule passes 2 electrons to the cytochrome C reductase complex, eventually that is going to reduce the complex, and the complex that is now reduced is able to pass it along the proteins of that complex to another mobile carrier called cytochrome C. Cytochrome C, it's soluble and it is in the outer membrane. It's part of the inner membrane, but it's in the outer leaflet of the inner membrane. And again, as the electrons are passing, they're going to be able to help pump protons. Eventually, the cytochrome C that has been reduced is able to pass 1 electron at a time to the cytochrome C oxidase complex. And that cytochrome C oxidase complex, the proteins within it are going to undergo electron transport chain reactions that eventually will lead to oxygen being the ultimate electron acceptor. So now protons are also going to be pumped in that complex. Not shown here, it's the place where you have the succinate coenzyme Q reductase. So ubiquinone is also called coenzyme Q. And there, at this step, this is the only step of the citric acid cycle that has a membrane-bound protein, and that enzyme, succinate coenzyme Q reductase, it's the one that is going to oxidize succinate to fumarate. In the same reaction, you're going to donate electrons, is going to accept electrons from FADH2 to generate FAD, and 2 electrons will be added to the quinone pool. But notice that there are no protons being pumped across, so only the NADH dehydrogenase complex, the cytochrome C reductase complex, and the hydrogen C oxidase complex are able to pump protons across the inner membrane of the mitochondria. Keep that in mind so you can then have an idea about the number of protons that are able to be pumped across the three systems, in whether it is NADH donating the electrons or FADH2 donating the electrons, because it's going to give you a different amount of energy and therefore a different amount of ATP. We're going to look at that later. So as the molecules are receiving and passing down electrons through the electron transport chain, you're going to have the pumping of protons across the inner membrane of the mitochondria. That is going to generate a proton gradient that we call the Proton-Motive Force. Now, in the first stage of that, you have the energy of the electrons transport that is now going to be used to pump the protons across the membrane, and as shown here in the left, the proton concentration gradient, the electrochemical gradient of those protons, it's greater in the outside of the inner membrane, so in the intermembrane space, than in the matrix of the mitochondria. So therefore, you require the energy of the electrons to pump those protons against their concentration gradient. Now, the second step, it's going to now harness the energy stored, the potential energy stored in that electrochemical proton gradient is going to be used to generate ATP via the enzyme ATP synthase. So this is what we consider the chemoosmotic coupling, linking the chemical bond formation of ATP to the membrane transport process via the electrochemical gradient of protons. And here on the lower left, I have put a YouTube link to a video that explains this very nicely. So when we look at the Proton-Motive Force, that electrochemical gradient of protons, we're going to have the same principles that we already learned about electrochemical gradients applied to the proton gradient as well. In here, you have the membrane potential within the inner membrane of the mitochondria. That it's described as delta V. The Proton-Motive Force uses a great amount of the membrane potential. So you see the arrow in red being very robust because there is a very large membrane potential in the inner membrane of the mitochondria. On the far right, what you're now seeing, it's the proton gradient or the pH gradient. So, the pH, as you know, it's the concentration of protons. So you can look at the change of concentration of protons across the membrane. So in the inner membrane space, we have a pH of about 7.2, and in the matrix, we have a more alkaline pH of 7.9. So it is a very modest concentration difference of protons across the membrane. Now, together, since the protons are moving preferentially to the negative charge within the matrix, the contribution of the membrane potential, it is significantly higher than the contribution of the proton gradient. So the electrochemical gradient of protons is the combination of the membrane potential and the proton gradient. So together they give a very strong drive for those protons to move across. So just to remind you, the Proton-Motive Force, it's the other name that we can use for the electrochemical gradient of protons. And you're going to see it used in bacteria, in plant cells, in their mitochondria, in plant cells, in their chloroplasts, and in every single cell that we can think of that needs to produce energy. Some bacteria, however, are able to use sodium gradients to also produce ATP, but we're not going to be discussing those more than what we just did. The last thing that I want to point out is for you to notice the different contribution of the membrane potential and the pH gradient here, because it is going to be significantly different in the chloroplast when we talk about photosynthesis. So keep this difference in mind, that in the mitochondria, the stronger force is going to be the membrane potential and not the proton gradient. And with that, I'll finish the lecture, and I'll see you later.